Hello viewers, today we are going to see about general knowledge biology. Topic heredity and evolution. The inheritance of characteristics through generation is called heredity. The inheritable characteristics are also known as traits. Each trait can be influenced by both paternal and maternal genetic material DNA. Georg Johann Mendel 1822 to 1884 worked out the first ever scientific experimental study on heredity. Mendel observed variations in the garden pea plant Pisum sativum. The contrasting characters that Mendel observed in the garden pea plants: stem height, tall, dwarf, flower color, violet, white, flower position, axial, terminal, pod shape, full, constricted, pod color, green, yellow, seed shape, round, wrinkled, seed color yellow green. Mendel termed the tall and dwarf plants as wild types or pure breeding varieties. In the monohybrid cross, Mendel observed all plants were tall in the first filial generation, F1. When F1 tall plant was allowed for self-pollination, both the tall and dwarf plants appeared in the ratio of 3:1. The first experimental of Mendel considering the inheritance of a single trait is called monohybrid cross. Expression of morphological characters is called phenotype. The expression of gene for a particular trait is called a genotype. The genotype of a character is influenced by factors called genes. The genes are the factors which form the physical basis for inheritance. The alternate expressions of the same gene are called alleles. The contrasting pair of alleles makes of an allelomorph. The differences in the characteristics among the individuals of the same species form variation. The variation that occurs in the body cells is somatic variation. Somatic variation is not inherited. Variations found in the germ cells or gametes are called germinal variations. Germinal variations is inheritable. Germinal variation leads to speciation and evolution. Variation is the source of raw material for evolution. Animals are able to adapt themselves to the changing environment by variation. The variation found among the individuals of the same species is called intraspecific variation. The variation found among the individuals of different species is called interspecific variation. The variation found among the individuals of different genera is called intergeneric variation. Sexually, reproduction produces offspring with marked, significant and visible variations. Variations give the organisms an individuality of their own. Without variation, there would be no signs of heredity. Charles Darwin put forth the law of natural selection. Natural selection involves struggle for existence and survival of the fittest. Variation leads to genetic diversity, which is the key for evolution. Evolution may be defined as a gradual development of more complex species from pre-existing similar forms. Evolution is an extremely slow process. Evolution has occurred over millions of years. Evolution is revealed by fossil evidences. Evolution has resulted in the diversity of organisms. Humans belong to a single species called Homo sapiens. Jean-Baptiste Lamarck, 1744 to 1829, postulated the use and disuse theory. Lamarck remarks that the will or want for a character makes the organisms to possess it at a later time. When two populations are isolated by geographical barriers, there is a chance for gene flow, genetic drift. Genetic drift leads to the formation of a new species. Genetic drift acts as an agent of speciation. Human evolution started before 15 million years ago in Africa. The early human-like creatures were called as hominids. 3 to 4 million years ago, hominids walked into eastern Africa. Hominids hunted with stone weapons and were mostly fruit eaters. Hominids were not taller than 4 feet and walked upright. The hominid was called Homo habilis. 1.5 million years ago, Homo erectus, who were meat eaters, lived on Earth. The Neanderthal man lived in East and Central Asia 1 million years ago. The Neanderthal man used to hide to protect them and buried their dead. Upsc Homo sapiens arose in South Africa. The Upsc Homo sapiens developed into distinct races in the Ice Age. Between 1500 to 10,000 years, the modern Homo sapiens arose. Prehistoric caves were developed about 18,000 years ago. Agriculture came around 10,000 years back. To understand evolution, evolutionary tree is used. Genetic engineering is the modification of the genetic information of living organisms. Genetic engineering is done by manipulation of DNA. Genetic engineering is also known as gene manipulation or recombinant DNA technology or DNA technology. The enzymes which can cut DNA into fragments are called restriction enzymes or restriction endonucleases. The enzymes which can join DNA fragments are called DNA ligases. Restriction enzymes or restriction endonucleases are the molecular scissors. Enzymes are biocatalysts that speed up reaction in cells. Antibiotics are substances produced by some microbes that help in increasing the immunity to human beings. Acetic acid is used for the production of vinegar. Edward Jenner, 1749 to 1823, in 1791, who coined the term vaccine. The first used vaccine was against hepatitis B virus. Vitamins are chemical compounds present in minute quantities in food. Dolly was a cloned sheep developed by Dr. Ian Wilmot. Dolly was developed in Roslin Institute in Scotland in July 1996. For the development of Dolly, nucleus of udder cell was used. The udder cell was taken from a 6-year-old fin dorsate white sheep. Vaccines act as antigens and stimulate the body to manufacture antibody. Steroids are a type of derived lipids. Example, cholesterol. Steroid drug Prednisolone is produced from fungus Rhizobus. Monoclonal antibodies are the antibodies produced by cloned cells. Monoclonal antibodies are used for the treatment of cancer. Cloning is a technique by which morphologically and genetically identical organisms are produced. The clone is an organism derived from a single parent by asexual method. Natural clones are identical twins. Induced clones or artificial clones are developed by nuclear transfer. The stem cells are the most unspecialized mass of cells. The stem cells can be induced to become any type of tissues with specific functions. Embryonic stem cells can be derived from early embryo. Vitamin B12 is biotechnologically synthesized vitamin. Vitamin B12 is used to cure pernicious anemia. The enzyme amylase is derived from amyloproteins of bacteria. Biosensor is a device consisting of immobilized layer of biological material and its contact with a sensor. Biochips are microchips which are developed by employing techniques of biotechnology. Biochips will be useful in defense, medicine, etc. Insulin-dependent diabetes is caused by the degeneration of beta cells due to a defective gene. Applying the principle of biotechnology, it is possible to correct the defective gene. Gene therapy is the means to treat or even cure genetic and acquired diseases like cancer and AIDS. In somatic gene therapy, the change is not passed to the next generation. In germline gene therapy, the change is passed to the next generation. If you like this video, you can subscribe my channel to get next video. Thank you.